Good morning, everyone. Thanks for joining us for day 2 of our CEPHaS virtual conference. For those that are joining again after our day 1 on Monday, thank you for coming back. For the new ones, this is our day 2. Our theme for today is soil properties and soil information. We have 2 external speakers who have kindly agreed to give us a presentation. There is Dr. David Chikoye from IITA and Dr. Matthew Mbanga from Foundations for Farming. And then we have 5 presentations from our CEPHaS team members. We will be starting shortly with David Chikoye's, Dr. Chikoye's presentation, and we are looking at having a break after 4 presentations at 9:35 a.m. GMT for 15 minutes or so, and we will join back at 9:50 GMT, as you can see on the screen. We are looking at finishing the conversation, the conference at 11 a.m. Thank you for joining us, and I'm going to pass the speaker to Professor Lark, who is going to introduce our first speaker. Thank you.
Thank you, Alessia. I'm sure most people attending the meeting know about IITA, the International Institute of Tropical Agriculture. IITA is a non-profit institution which undertakes research to generate innovations, agricultural innovations, in Africa to meet needs of food security and environmental protection and poverty. Dr. Chikoye is director for the regional hub of IITA for Southern Africa and the plant production and health management thematic area. And as you can see on the screen, he has a particular interest in weeds and their place in agricultural systems, management options from herbicides to the use of cover crops and other such systems. So we're very much looking forward to hearing Dr. Chikoye's talk and thank him for his time to speak to us this morning.
Right, sure. From the beginning. Okay, thank you very much for giving me this opportunity to talk to this group. My name is David Chikoye, as already introduced by the chairman. I'm the director IITA Southern Africa, as well as plant production and health management. Now, what you see in the pictures here, some of our stations, the middle one, this is IITA Zambia. Then the next one is, the one down is IITA Mozambique. The top one is IITA Malawi. Can somebody confirm? Can you see me? Can you hear me? Sorry.
Yes, we can. Thank you.
All right. Very good. Okay, so my presentation outline is as follows. Just brief comments on the CGIAR network, then delve more into IITA, the research portfolio. Finally, since this group is about talking about conservation agriculture, I thought that it was going to be important for me to share some of our experiences on the study we did on adoption of conservation agriculture. Now, moving on, what is the CG? The CG stands for Consultative Group on International Agriculture Research. This is actually a largest network of centers who are involved in global agriculture research. Their main focus is on reducing poverty, enhancing food and nutrition security, and improving natural resources. So from the picture here, you can see where the different centers are located. But what is important to mention here is that IITA is African-based, located in Nigeria. That's where our headquarters is, but we operate through 5 regional hubs, of which Southern Africa is one of them. When you look at the original mandate, we had a breeding mandate for 6 crops: soybean, cassava, maize, cowpea, and other crops. But also, more importantly, we had a farming systems mandates. where the main focus there was trying to look at systems that can improve slash and burn. Somehow. All right. So more about IITA. IITA generates agriculture innovation to meet Africa's most pressing challenges of hunger, malnutrition, poverty, and natural resource degradation. And since about 10 years ago, we started a program on youth in agribusiness. IITA is present in 35 countries in sub-Saharan Africa, where we work with numerous national and international partners. Now, since our focus is on smallholder farmers, we need to look at the characteristics of these smallholder farmers. Number one, they are land constrained. Two, they incur high post-harvest losses. They have limited access to input and output markets. They get liquidity constraints, especially at the time of planting, and also have limited access to credit. They incur high transportation costs, given that they do their activities in remote areas. They face unpredictable agriculture policies, face unpredictable weather. Talking more about this, today is almost the second week of December, where everybody should be in the middle of planting. Unfortunately, rains have not even started. This group of farmers experience low productivity. As a result of all these constraints, all these farmers are classified as poor. Now, again, if you look at agriculture in Africa, it faces multiple challenges, which can broadly be classified as biotic as well as abiotic. The abiotic stresses include heat, drought, poor soil fertility, and many others. Biotic stresses are many. I classify them into two, the old ones, like the parasitic and non-parasitic weeds, diseases of cassava, diseases of bananas, diseases of soybean. But we also have emerging biotic stresses, such as the fall armyworm, maize lethal necrosis, banana Panama disease, and then Electra in legumes. Also to mention here that, farmers, they use suboptimal crop management practices to address these challenges. Now, at IITA, what do we do to address these challenges? We are into maybe 5 main things. We are into research, where we generate technologies, tools, protocols, practices. We also facilitate technology deployment. We have a large program on capacity development. We don't confine degrees, but we support graduate studies. Graduate students who are actually assigned to our projects, they do their research with us, and then they go back to they complete their degree at the university. We do offer advisory services, and also we do a lot of information sharing. Now, what we do, we work a lot with partners. For research and technical support, we work a lot with fellow CG centers. We work a lot with universities. We work a lot with advanced institutions. Here, what actually we are trying to do is mobilize technologies. Then in terms of advocacy and political support, we work with a lot of national, regional, and international organizations such as the African Union, COMESA, CADESA, FAO. Then we work a lot with the national government, like in the area of variety release. Then we work a lot with farmer organizations who are actually the link to the ultimate beneficiary of our technologies. Then of late, we've been working a lot with the agribusiness, especially in the area of seed commercialization. Now, apparently there is one slide which is missing, where I had outlined the main research areas for IITA. We do a lot of agronomy. We do a lot of pest management work. We do a lot on food science and nutrition, natural resource management, and then we are working a lot on developing seed systems. So I'll briefly give you a few maybe more detailed examples of some of our research work. The first one will be on the work we've done on 4-armyworm. 4-armyworm was discovered in January 2016 in Nigeria. By the end of the year, 2017, it was confirmed in 44 countries. Now, what is special about this worm? The host plants are many, over 100 species in 26 countries. And in a place like Africa, where we do a lot of intercropping, this has a lot of implications for our farming systems. The preferred crop is maize, though many other plants can be attacked. Again, the issue here in Southern Africa, maize is our main staple crop. So when the problem arose in 2016, we needed to have short-term interventions to control the pest because, you know, farmers wanted to make sure that their food security is not compromised. Then we needed also medium and long-term solutions. So in IITA, globally speaking, what we do, we try to encourage integrated pest management, where the first entry point is doing surveillance as well as early warning systems. So farmers actually should go and scout their fields. If they find that there is a problem, this is when they need to intervene. And about 5 years ago, the most used intervention was pesticide options. We had no other options because the pest was new. Then while farmers were using pesticides, there was research which was focusing on culture and mechanical control, biological control, developing maize cultivars which are resistant to the pest, as well as there was work on policy, especially as it pertained to herbicide use. And then also what we were doing as IITA, we were mobilizing knowledge. It was a new pest here, but an old pest of the Americans. So we didn't need to reinvent the wheel. We needed to look at how they control the pest here and then start adapting it for our environment here. Climate change has become a topical issue and it's a reality now. 10 years ago, we were predicting climate change, but now what has happened is real, it's living with us. So what is IITA doing? We have focus on developing climate resilient crops, especially crops like cassava, cowpea, and bambara nuts. Naturally, those ones are more drought tolerant than maize. Now for maize, which is our staple crop, and other commercial crops like soybean, they actually deliberate breeding efforts to breed more crops which tolerate drought. Also, we are promoting crop diversification to increase mitigation and adaptive capacity of farmers. Again, here I'm saying that let's bring more cassava and cowpea in our traditional crops. On the agronomy side, there are a lot of things we're doing like timely and optimum planting, maize legume intercrops, maize legume rotations. We are looking at water management options. We are looking at issues around conservation agriculture. So the focus of this meeting is looking at the biophysical aspects of CA. But what I wanted to do here is to share with you some of a recent study we did to identify the drivers and barriers of CA adoption in Zambia, Malawi, and Mozambique, and Zimbabwe. But today I will mainly talk about some of the key results coming from Zambia. We have evidence that CA has demonstrated positive agronomic as well as economic benefits. And these are not just studies done by scientists, even the farmers themselves, they talk about these benefits. So over the last 3 decades, there have actually been a lot of investment by donors and national governments to address low agricultural productivity through promotion of CA. But unfortunately, the adoption has been low, maybe below 10%. So the objective of this study was to identify the drivers and barriers to CA adoption. The work was done in 7 districts, and eventually we interviewed 1,400 households. Now, some of the key results. Farmers were highlighting declining soil fertility and droughts as the main constraint to maize. Remember, CA is mostly promoted in maize. So when we promote CA on a biophysical aspect, the question is it actually addressing soil fertility as well as drought, which is the main concern for farmers? We also found that there was a lot of awareness, but low adoption of CA. So for example, if you look at minimum tillage, 97% of the farmers were aware of CA or tillage, but there was only 53 adoption. Mulching, rotation between 80 and 92% awareness, and again, lower figures for adoption. Look at the full CA adoption. 45% awareness, about 33% adoption. Now, also, some of the results we are seeing are that there are more farmers who are disadopting than adopting CA. Look at minimum tillage, where you find that, let's say last year, 8% were testing CA, while 14% were disadopting. These same results are similar, whether you're looking at mulching, rotation, intercropping. So this suggests that, adoption of CA may decline over time because of high rates of disadoption. Then also what we were seeing was that there were many channels for promoting CA, but then it seemed that demonstrating plots were the best for promoting CA adoption. And this is understandable because CA is a combo. So technically, it could be challenging actually to disseminate it through other mechanisms. Now, what were the main reasons for non-adoption or disadoption? Quite a lot. Some had a bearing on lack of credit, limited technical knowledge, high cost of fertilizers, limited support from public input distribution system, high labor costs, limited crop residue, lack of incentives, lack of supportive institutional bylaws. I think this is pertaining to the use of mulches, and then unavailability of vacuum seeds. So in terms of summarizing this work, where we see the key barriers, these are lack of technical knowledge, then poor access to complementary inputs, labor constraints, especially for weeding, and these issues around crop residue management. So let me end here and entertain some questions if there are any. Thank you.
Thank you, Dr. Chikoye. Thank you very much. We're just going to check if there are any questions for you. I think Murray may have a question. I am just going to ask Murray to speak, please.
Thank you very much again, Dr. Chikoye, for a very interesting talk. When you were talking about factors which are disincentives to adoption of CA, I was wondering about the significance of weed control, because of course cultivation is one way of achieving weed control at the time of planting. And given your particular interest in weeds and their place in systems, I was wondering whether you had any views as to the way forward for making CA more attractive to farmers from the perspective of improved control of weeds, or dealing with weed control as a barrier to adoption.
Okay, Mr. Chair, can I respond?
Please do.
All right. You know, we are actually extensively discussing this finding. A few things we need to discuss. First, in CA, the way it is promoted in Africa, 2 herbicides are mainly used for a burn down treatment at the time of planting. Glyphosate and paraquat. Now, these herbicides have issues. There are health issues. We could also potentially be promoting the emergence of weeds which are resistant to herbicides. So this is one area we need to look at, that, you know, at the time of planting, what options are available for farmers to use? Because you see what, already we need to get into weed resistance management before it appears. Now, also farmers actually were saying, remember that, you know, CA, you start getting all these benefits being talked about, maybe 5 years. It's not in the short term, you know, because over that long period of time, the idea is actually to reduce the seed bank. So if you reduce it and then it's just maintained in the first 1 centimeter, you know, soil depth, you know that over time, then the weed pressure gets low. So again, the issue here is that we will get rid of most of the annual weeds, but perennial weeds are going to increase. But the main issue for farmers is that weed pressure gets high in the formative years of CA. And the question is, they don't seem to have many options for controlling those weeds because farmers intercrop maize with other crops. So I think that is the main issue. So in a scenario like that, probably herbicides are not going to get, they're not going to be more prominent. We need to look at other options for weed control. So maybe let me end here. But for me, these are the 2 serious issues: preventing herbicide resistance, getting options which can reduce the labor burden during the formative years of CA, and then looking at options beyond herbicides in areas where farmers do a lot of intercropping. Thank you.
Thank you very much.
Thank you, Dr. Chikoye. I think there may be another question. Shepherd, would you like to come in?
Yes, thank you. Thank you very much, Doc, for the presentation. I was looking at some of the reasons in profile for, you know, this adoption of CA, and I see that the things like lack of fertilizer and so on, which to me is a bit intriguing, because I think if farmers leave CA, they go back to their old practice, which is conventional. Perhaps as an explanation, maybe there's a requirement that CA is required, requires fertilizer, and perhaps conventional methods that don't require fertilizer. I think there's something interesting there you could explain further, Doc.
Thank you. So this is for fertilizer. There are many issues here. Remember, farmers have said that poor soil fertility is a constraint, and they do believe that CA has a role in improving soil carbon. Now, if you look at the way CA has been promoted in all these experiments, in all these demos, you know, yes, CA is about reduced tillage, mulching, and intercropping. And farmers somehow by default have also been receiving fertilizer. So it's like for farmers, fertilizer seems to be the 4th pillar of CA. And if you talk to farmers, they will actually tell you why they cannot even expand beyond this CA demo. They always tell you that, okay, look, the fertilizer I received cannot be, you know, able to support an area beyond this CA plot. If you look at farmers in Malawi, they practice tillage, you know, they form ridges. But look at what they do before they form those ridges. They collect all the remaining vegetation, they put it along the furrow. Then they will break this ridge on the left and on the right and bury this mulch. Why do they do that? Farmers do believe that, you know, what they're doing there is adding organic matter. So even though they plant without fertilizer, that organic matter is a source of nutrients. But remember, in CA, we are saying that no, don't what? Don't do any tillage and leave this, the crop residue on the surface. That crop residue you leave on the surface, one, you lose it to animals, you lose it to fire. So it doesn't give that benefit of nutrition. So for me, it's not really about, okay, this is the, remember we are reporting on farmers' perception. So maybe that explains why farmers feel that, look, fertilizer should be part of the component because the traditional system they use now is being challenged. So there has to be another option to make sure that crop nutrition is addressed. Thank you.
Thank you, Dr. Chikoye, for answering that question. One last one, please, from Dr. Richard Owen at UZ. Is, do herbicides have a negative impact on soil biota and then downstream on soil structure?
Sorry, I didn't understand the question.
If you, can you understand me? Can you hear me properly?
Yes, I can hear you.
Do herbicides have a negative impact on soil biota and then downstream on soil structure?
Okay, on soil structure, I am not aware of that. Some herbicides, yes, may have impact or negative impacts on soil biota. A long time ago, actually, we did a study where, for example, we are looking at the impact of some of these, you know, soil applied herbicides on, you know, inoculation. And apparently a wide range of those herbicides did not show any negative interaction. So it depends on the kind of herbicide you're using and also on the kind of soil biota, but that work continues. Thank you.
So much. Thank you. I think we are done with the questions and it's time to move on to our next speaker. So thank you very much, Dr. Chikoye, for your time. We really appreciate it. And I'm going to ask Murray to introduce the next speaker now, please.
Thank you, Alessia. So our next speaker is Dr. Craig Sturrock from the University of Nottingham. Craig is presenting on behalf of colleagues at Luana, University of Zimbabwe, and Rothamsted Research. And Craig will be talking about the use of X-ray computed tomography for examining soil structure under conservation agriculture. So thank you, Craig, and over to you.
That's great. Thank you, Murray. Can everyone hear me okay? Yeah.
Yes, thank you.
Like that. Thanks, Alessia. Okay, good morning, everyone. So as Murray introduced me there, my talk today is going to be using this technique called X-ray Computed Tomography to look at soil microstructure in some of the field trials in Malawi and Zimbabwe. I've been using the X-ray imaging technique for probably about 12 years now, while I've been based at the University of Nottingham, but also working back in Scotland when I first started using it. So the key thing really is, we're looking at soil properties today, talking about soil properties and soil information. And perhaps one of the most important soil properties is relates to soil structure, and it's fundamental to how a whole range of things really. And if we start talking about soil structure on soil function, as scientists, we might think about it in different ways. So the soil physicists might be concerned about sort of water, movement of water and gas particles and soil particles through the soil profile from the surface into the deeper layers of the soil. Soil chemists might be more interested in nutrients related to soil fertility and also contaminant movement through soils. For the soil biologist, it's really the sort of the habitat space, all the different soil pores there, how connected they are, habitat space for microbes, and obviously you've got also larger organisms in there, micro invertebrates, and also the important plant roots moving through. So the fundamental sort of architecture that describes the soil structure has impacts in all these different areas. But moving away from sort of science, it's really thinking about the farmers. So how good structure in soil, these benefits the farmers with improved drainage. There's going to be reduced soil erosion from excess surface runoff, and you've got a much more better aerated soil. So there's less anaerobic microbial growth and things in the soil. Obviously, you've got unrestricted plant root growth is a key thing. If you've got compacted layers where you've got much denser region, it's sort of you're going to plant roots are going to struggle to get through those root layers, and ultimately having better yields. So measuring soil structure is traditionally been a bit of a black box, really. You tended to dig up the soil and have a look at the sort of soil pits and try to maybe count roots or look at the pore structure as you move down from the soil surface into the soil. The other way is really taking soils back to the lab and really sort of disrupting it and breaking it apart and putting it into little cores and measuring different properties about them indirectly. But really what the problem is doing that is that you're actually destroying all the structure that's in there. So there's a really sort of drive for a more non-destructive method to be able to do this. And that's where this X-ray imaging technique really comes in. So it's first used in about the 1980s to start looking at soils. And the technique's really sort of developed over that sort of 40 years now of getting used in the sort of soil sciences areas to these sort of modern systems. But the technique's really fundamentally, it uses an X-ray source inside one of these sort of CT scanners, and this is the one that we have in our facility at University of Nottingham. And inside here we've got an X-ray gun, somewhere to put the sample. Here we've got Tongai from University of Zimbabwe when he visited in 2019 to do some training, placing one of the soil cores inside the scanner cabinet. So the X-ray beams pass through the soil core and we collect a series of images on a detector panel as the samples rotated round. And then the output images are these non-destructive cross sections through the image. So the one here, the 2D slice, is kind of right down through the middle. I'll talk about the analysis in a moment. And then we can also get these sort of three-dimensional renderings of where all the pore space is inside these cores. So the key benefit is that it's completely non-destructive. So we can scan these cores, then they can be sectioned up and then done, either scanned at a higher resolution or different chemical or other biophysical properties measured on exactly the same cores. So the structure of the data it's all these sort of grayscale images, and really the color intensity here, so going from the darkest color values in the images up to the brighter bits, relates to what we call the X-ray attenuation. So it's basically the density of the material. If that's higher, there's more X-rays going to get absorbed by that material. And this is reflected in this histogram here, going from just the number of pixels in that image that are represented at low, darker regions up to the brighter regions that are high densities. And obviously, the output is, when we take one scan, is probably a couple of thousand individual images as we move through. So this is dictated by the resolution of the scan. So here, we're probably working at about 50 micron resolution. So each image in this sequence here is a 50 micron slice through that soil. So the amount of data that we generate is it's massive, really. So it's really obviously informed. You've got that digital data set that you can then move through to start quantifying things that's in the cores. We can also view it in 3D. These are just some images as we slice through that core. And as we zoom in, we can start to maybe pick out some features in there qualitatively. So there's a sort of piece of organic material here, probably some plant residue that's been incorporated in the soil. You can see some smaller stony regions, and then you can see different cracks and perhaps sort of aggregate structures in there, as well as sort of some void regions where you've got biopores going through that soil. And really, as we dive in a little bit deeper, what I've really found fascinating working with soils on CEPHaS project is the amount of really interesting structures you see in the soils. In working with a lot of the UK soils, we often don't see a lot of this sort of macro and invertebrate activity that we can see in the soils from the CEPHaS project, where you see a lot of these biopores that have maybe been infilled. You sometimes get these little sort of cemented structures that perhaps some some insects have been creating. So they're really fascinating to look through. Here we've got some biopores in the center. And if we zoom in here, there are actually some plant roots that grow through the biopores. So the key thing is really, if you've got well-connected soil structure with these deeper biopores that go into the soil profile, they perhaps give a path of least resistance for plant roots to move through. So having this sort of nice connected soil structure throughout the soil profile is beneficial for plant growth. Image processing is a large part of the project. So obviously we've got to take the soil cores first, then they're scanned, but really the major bottleneck in the data is being able to process these images. A typical soil core that we took on this project are about 30 centimeters long, and you get roughly about 6,000 to 7,000 individual images like this one on the left here, this XY slice, per sample. So it takes a long time to actually work way through and process it. So typically the sort of image analysis flow through, we've got the stack of XY slices. Then we take a smaller region of interest from here to sort of crop it down to remove any edge effects. And then we want to start separating out where the solid material is from the pore space. And this is done using this technique called thresholding, where we binarize the image. So all the dark regions here are all the pores. And then we use the software to be able to count up the number of pores, which in the pore area, which then allows us to calculate things like the soil porosity. Obviously, because we've got it throughout the full length, we can look at the depth versus the porosity. We can look at pore number, size, surface area, shape. And when we extrapolate this out into 3 dimensions, we can then look at the connectivity of the pore structure and also how sort of tortuous it is. So it's gives you an idea of the kind of the wandering path, really. It's whether the pores are much more direct or they're kind of moving, taking a kind of longer path as they move down. So within the CEPHaS project earlier this year, we took a number of cores from the field sites in Zimbabwe and Malawi. In Zimbabwe, there were 2 treatments that we looked at. There was a conventional tillage and a no tillage treatment with some residues added. There was 3 replicate blocks, and there was 3 replicates from each block collected. So we had a total of 18 cores. The cores that we analysed were all from the top 0 to 30 centimetres of soil depth, but there were additional cores taking it down to 1 metre that may be analysed in the future. In Malawi, we looked at 3 treatments. So we had a control, conventional agriculture. We had then 2 CA treatments, one with maize with direct seeding and dibble stick, and then we have a maize intercrop with legume. Again, 3 replicates, and we had 4 samples from each block. So we're just going to really look at some of these images now. This gives you some examples. So this is the conventional tillage treatment within Zimbabwe. So what was interesting about these, the soil was actually, there wasn't a high number of biopores in here when you were looking at it. The soil was actually quite condensed, kind of looking through them. So as we go down, we've got a sort of front view of the soil core showing the 30 centimeters, and then I've just put up 2 representative slices through. That's about sort of 60 centimeters, 6 centimeters down, and then about 120 or so. So yeah, we can see these images. Comparing them to the no tillage, again, what's really prevalent is the number of cracks that you can see down the cores. Now, unfortunately, I think probably due to the transit from the soil cores from Zimbabwe all the way to the UK for doing the imaging, there is probably some disturbance. So unfortunately, quite a lot of them have these cracks going through them. So if we start looking at the soil structure here, we've got some of the replicates that are really nice, intact structure. They don't really have any of these cores that's gone through, but many of them have these sort of cracks running through, which is kind of complicates the analysis, shall we say, because when we start looking at the numbers, once we go into the next slide, so what we've done here is just look at the soil porosity with depth. So we've got 0 to 10, 10 to 20, and 20 to 30 zones, and comparing the porosity in the different zones. And really, I couldn't find any statistical differences in the porosity between the 2 treatments. And I think really the main explanation for this is due to this higher number, the sort of prevalence of these sort of cracks down through the cores. So we just look at the first conventional agricultural treatment. If we look at the 3 replicates there, we've got 2 that didn't have cracks and 1 that did. And if we plot these with depth, you can see that the middle one, the R2 there, does have this much higher level of the cracks. And we can see that plotting that. So really, I need to work out a different way to approach how to do the analysis. And obviously, because the data it's all there digitally, we can just sort of subsample it. So the idea that we have for this is just to look at smaller regions of interest across all the treatments that avoid areas where we've got the cracks, and then start to plot this through. Another alternative is that because we've still got all the cores, we can actually section them up, cut smaller regions out of them from the top, middle, and bottom, and we could actually maybe scan these at much higher resolution, which maybe just give us a bit more information, because the sort of denser soil structure here, scanning at higher resolution is going to reveal more of the sort of microstructure in those pores and maybe show us some nice differences. So this is the data is very much still in getting processed at the moment, so it's ongoing. For the Malawi site, we've got again just a few representative images. So the data is still in progress with these analysis. What's interesting, just sort of comparing the Zimbabwe soil and the Malawi one, you can see quite a big difference in the soil structure. The Malawi ones has these sort of quite large, sort of blocky, solid, aggregated structure interspersed with a finer, more open pore structure. So yeah, it kind of highlights the difference between certainly the soils in the 2 countries. Just a quick look at some of these again. That sort of heterogeneous sort of aggregated structure is prevalent throughout the replicates. If we I've just plotted one, the porosity here, and we get a slightly higher porosity as we move down through those. And again, just kind of looking into those soils, you can kind of see these. So there's definitely more evidence of larger biopores here. You can really detect them by these sort of circular shaped ones as we go down. So once we get the analysis finished on these ones, they're going to be quite interesting. This one here within treatment 3, one of the conservation agriculture, you can get an idea here of the cemented walls you get in some of these biopores that I think are perhaps engineered by insects, by ants or termites perhaps. And again, the final treatment there. So I say the analysis is still in process of these ones. We're working on that. Should get them in the next few weeks. So really, just to wrap up, I think the X-ray imaging was certainly successful in measuring the soil structure in the cores and CEPHaS project. But obviously, the image analysis is still in process to fully characterize the soil pore structure. But we're going to, obviously, we've got a few challenges to fully classify that. So we're going to use maybe aggregate scale scanning to complement existing data set. And the key thing is really, the final thing is being able to complement the soil structure data from the X-ray imaging with the other measurements that we've got in working group 1 for the water retention data, or the water infiltration, and the soil moisture data. So bringing all that together is going to be really important. I'd just like to finish by thanking everyone involved in the project, and I'm happy to take any questions. The image here was from our the workshop that we ran in 2019 with our colleagues on the project. So thank you.
Thank you, Craig. That was a very great presentation, looking at another angle that CEPHaS has managed to study as part of this collaboration. So thank you. I can't see any questions in the Q&A at the moment. I'm not sure if anything has been posted in the chat. I'm just going to give people 1 second. Okay, I don't think I can see anything.
Okay. If I could just ask a quick question, Craig.
Yeah.
Can you say anything more about the kinds of quantitative analyses that you think might be useful to apply to these scans in order to get some insight, particularly into what differences there might be in the sort of porosity associated with macrofaunal activities, termites and so on?
I'm still, so I think it's from the actual images, it's quite difficult to separate out where the origins of some of these pores have come from. So some follow-on work that I've been doing with Kate Parr at the University of Liverpool. She's interested in termite activity, and really felt I met via Martin Broadley, actually was introduced from sort of follow-on work from CEPHaS, but she's interested in termites, and we took some cores from South Africa where there's a high level of ants and termites in these soils. And there's some really fascinating structures inside the images, but what's difficult is that we don't know how long they've been there. So you get these sort of structures that they've almost got a really high density. It's almost like the walls of the pores have been cemented by some material, and then it kind of, there's a lower bulk density behind that region. But because we don't know what the soils were like when we started, we don't know what's created them or how long they've been there. So we've been talked about building up some new experiments where we actually dig a soil pit, remove all the structure by sieving the soil, repack it, and then take cores and then do a time series, like a number of steps afterwards to look at, see what we can see. And they do these sort of exclusion experiments where you can knock out either an ant or termite population and maybe look at the differences in there. So I think perhaps a method like that may help inform some of the origins of these, but it's challenging to pull out the structures just on these binary images. I think you get a lot more information in these grayscale values, but there's not many methods to actually look at it yet. So there's lots of work. It's still kind of an emerging technology, being able to identify what these things in the images.
Just one question, which is in the chat from, I think from Lydia. She says, were the differences in soil structure really due to inherent differences in soil type? And she asks whether, to some extent, this is, you've addressed this in what you've just said to me, but whether you'd found any differences in mesofauna and root biopores.
So hopefully we'll find more differences. It's very much the data is still getting processed, especially the cores that in Malawi, really. We've done all the scans and we're just sort of moving through the actual quantitative analysis, really. So it's kind of a multi-phase process. It's a case of we generally process them all, see what the numbers look like, and then sort of more go into sort of targeted regions to see if we can get them through. So there are differences in certainly soil type, I think, between the Zimbabwe and the Malawi cores. There's also a big visual difference you can see there. So whether that's down to the underlying structure of the soil, that we're looking at, I think, a sandy loam to a sandy clay loam. And, so yeah.
Thank you very much, Craig. There are some other questions in the chat, but we are going to have to push on. So hopefully you'll be able to see those, or we'll be able to make sure that they're passed on to you, and we can share your responses. But that'd be grand. Thank you very much, Craig.
Thank you.
Our next speaker is Chikoye Miti. Chikoye, formerly from the University of Zambia, which is where he's been working on the CEPHaS project, but is now doing a PhD at University of Nottingham. Chikoye is going to be talking about pedotransfer functions for Southern Africa, work done in the CEPHaS project to evaluate statistical models to predict soil hydrological properties from routinely measured soil physical properties. So I'll hand over to you, Chikoye, and we look forward to hearing your talk.
Okay, thank you very much, Mari, for that introduction. Alexia, could you please start the slide? Sure. Okay, so I'm going to present on works that we have done in the project concerning pedotransfer functions in Southern Africa. So this is going to be a review and quantitative appraisal and reparameterization of selected pedotransfer functions. And this is a joint work between Working Group 4, which is a statistics working group, and the soil physics working group. Next slide. Next slide, Alison. Okay, so as an introduction, we've heard from the previous speakers from today and some who spoke on the Monday session, talking about how climate change is projected to affect yields. And unless we come up with intervention measures, measures that deal with soil and water conservation, there will be impact on yields due to climate change. So for us to come up with these measures that help conserve water and soil, there's need for us to have knowledge on the soil physical and hydraulic properties, and that will help us to come up with more effective measures to counter the climate change. Next slide. Yeah, so what then is the problem? So for Southern Africa, particularly Zambia, Zimbabwe, and Malawi, there is missing hydraulic property data in most databases that are there. So this has hampered the development of more effective soil, water, and soil physical management practices. mainly because these soil physical and hydraulic properties are difficult to measure in the field, and also because there's generally a lack of equipment to carry out these measurements, and more so that to do these measurements, it takes time, it's more time consuming. For example, for you to do the soil moisture characteristic curve, for example, it takes more than 2 to 3 months for you to come up with a complete set of measurements. But not all hope is lost. There is a solution to this, and one thing that we can do is that we can try to predict these hard to measure soil properties using other properties that are available at our disposal. For example, you find that in most reports, there are measurements done on soil particle size distribution, organic matter, and bulk density. And these can easily be used to predict other properties like hydraulic conductivity and soil water retention by the use of Pedotransfer functions. So despite now having these functions, the Pedotransfer functions at our disposal, they have not really been used, fully utilized in Southern Africa to fill in these missing gaps of soil hydraulic properties, which is why we undertook this study to try and evaluate the performance of some of the existing Pedotransfer functions, both from the tropical region and from the temperate region, to predict soil water retention in Zambia, Zimbabwe, and Malawi. And furthermore, using data from these 3 countries, try and come up with functions which are specific for soils of these 3 countries. So to do this, the first task that we did was to collect soil data from the 3 countries. So we collected soil data on different soil properties, particle size distribution, organic matter, bulk density, as well as volumetric moisture content, altitude capacity, and wilting point. So we went into the libraries of these different countries, checked soil survey reports, scientific papers, PhD thesis to extract this data, and you can see the map on the right, showing the points of where this data was collected from. And the way we collected this data was that for each entry, for each data point, we had to record the horizon from where it was collected, the profile where it was collected, and the site from where it was collected. So we had different codes for these 3 levels of collection. Yeah. So after collection of the data and compiling, we had to select some of the commonly used Pedotransfer functions in literature. So both from tropical and temperate regions. So the ones in blue on your left, they are Pedotransfer functions from developed in the tropical domain, and the ones in green, functions which were developed in the temperate regions. So to evaluate these functions, we use 3 criteria. So the first one is that we did some scatter plots of the predicted values against the measured values, just to see how they relate. Then we also did the mean error, which is basically the difference between the measured and the predicted values. Then one other thing that was done was that we tried to check how these pedotransfer functions perform in terms of in space, but as well as with depth. So for every measurement that was done, which is the Z in this equation, which you see down there, it was a function of four components. So on the first component on the right is just basically the mean value of that measurement, or due to the factors, the soil factors which were there, like clay, silt, and organic matter. So the X is basically just a vector of coefficients, and the beta is also a vector of soil properties. And what you see, everything else on the right, is the random effect. But we also split the random effect into three components. So the first one is the random effect, which is associated with a measurement being in a profile, in different profiles, which are separated by a distance. So it's like the between site random variable. So then the next one is the random variable that is associated with profiles that are within the same location. Then finally, the last one is the random variable that is associated with measurements within the profile. So the preliminary results that we have so far, when we look at the results at field capacity for these predictions, on the left here, we see four scatter plots for the predicted values versus the measured values. So what we discovered was that there were three distinct categories that we had. So the first one in the top left corner is a category which we had functions which had good relation between the predicted and the measured values, and they agreed almost very well, and they were close to the one-to-one line. While there were other functions like the one on the top right and bottom right, the measured and the predicted values related very well, but they were either above the one-to-one line or below the one-to-one line. So these were classified as biased, either they over-predicted or they under-predicted. But there was also another group which did not have a good relationship between the predicted and measured values. And like the one on the bottom left corner, the relationship is not quite good, and we classified those ones as no relation. But coming to the main criteria that we used for evaluation, it was we used the sum of the variances that I talked about in the previous slide and the mean error. So the idea was that if a function has a lower sum of error variance and a mean error that is close to zero, it was considered to have performed good comparing to the other functions. So you can see on the right there, there's a plot of sum of error variance against the mean error. So just the next slide, Alicia. So we can see that there are two, on the bottom there of the plot, there are two functions which did very well. One of them is Botula 2012. It was developed in the tropical region, somewhere in the Congo, as well as the Minasni and Hatemi, also a tropical function. And one other thing that was noticed was that the functions from the temperate regions, in this case, did not perform very good. An example is the one in the far left, top right, top left corner there. Yeah, one of the temperate functions. It had very high sum of error variance, as well as it was deviated away from zero in the negative side. So that was a downside of the temperate functions, but nonetheless, not all of the temperate functions performed badly. One particular one is the Saxton and Rouse, which performed relatively better compared to other tropical functions. And this is good because this function is usually found in most water transport softwares, which are used currently. So it's a good sign. Next slide. Yeah, then coming to the water retention at Wooting Point, almost the same story. If we look at the scatter plots, here I just have two scatter plots. One for a function in the temperate region, the one by Rouse, and the other Vandenberg, tropical Pedotransfer functions. So what I just want to show here is that we can see that this function from the temperate region, the Rouse, it did not perform very well as well at this tension. As you can see, it had a very high range of predictions for a very narrow range of actual measurements. And some of the measurements were actually unrealistic. For example, you find that it would predict a moisture content of about maybe above 60, 80, or even to 100 sometimes, which is unrealistic. But however, for the tropical functions, at least they were able to predict within the range of realistic measurements. So, and they were very close to the one-to-one line, showing that they did perform very well. When we look at the back. Yeah, when we look at this, the slide, the plot for the sum of error variance and the mean error as well. Same story as previous with the field capacity. We have a cluster of tropical functions down below the plot with a low sum of error variance and close to the mean error close to zero. And again, the function of Botula, as well as Vandenberg and Jickerman, all tropical functions were seen to perform very good. And again, the temperate functions did not do very well in this at this tension as well. Next slide. Yeah. So the other thing that was done, like I previously mentioned, wanted to see how these functions perform both in space and in depth. So the sum of error variances were decomposed into 3 components: the within profile, within site, and the between site. So if we take, for example, the function that was consistently performing well, the Butola function, we can see that it's the one with the triangle pointing down. We can see that at the within profile variance, it had a lower variance compared to the within site and between site variance. So this shows us that it was able to predict well at profile level than at broad scale level. And this is particularly important because most of the studies involving water transport are done at profile level. At rooting point, a slightly different story. Again, if we look at the same function of Botula, this time we can see that the variance at within profile is low, but it's also comparable to the variance at the between site component. Yeah, just the last one here. Yeah, so here it tells us that at rooting point, both profile and broad scale predictions are good for this function. Next one. So that was with the evaluation of the current functions, but what we want to do next is that we want to develop functions, Pedotransfer functions specific for Zambian, Zimbabwe, and Malawi source using the data that we have collected. And how we aim to do this is that we want to, first of all, want to create functions that are have different levels of input, such that if you do not have one particular input, there should be a function that is available for you to use with the inputs that you have. And we aim to do this by using a linear mixed model so that we appropriately deal with the clustered nature of the data that we have. And this is in progress. One exercise that we've done already is that we did carry out an illustration exercise just to have a feel of what experts expert opinion is on which soil properties are more important in prediction of soil water content. And we have received those questionnaires, and we're in the process now of coming up with new functions, which will be ready in due course. So this is how far the study has gone, and we hope to complete in a few months' time. So thank you very much for your attention. I rest my case here.
Thank you, Chikoye. Thank you. There is one question for you on the Q&A. I am just going to put it up. Seems Lal and McLean are not fitting the data well. Is it not sensible to fit a quadratic function for your Pedotransfer function? So I think this refers to when you were presenting this slide.
Yeah.
Can you see the question? Have I read it well?
Yeah. let me just read it. Okay, yeah. So these are functions which were already developed, and we tried to fit them to our to the data that we have, so that we see how they perform. But as we are going to be developing new functions, we're going to see how they fit to the data and be able to decide which function to fit to them, whether it's a quadratic function or other functions that will fit in the data very well. But for these ones, they are already existing, and we just wanted to see how they are able to predict.
Thank you, Chikoye. And just to add to that, I mean, some of those functions we examined did have quadratic terms in them, and they didn't necessarily behave any better. Of course, you're asking for trouble sometimes by putting higher order terms into a function like that, particularly if you range into areas of the parameter space outside your original calibration set. Thank you very much, Chikoye. That was a very clear and interesting presentation. If there are any other questions, we'll be able to pick them up through the chat, but we need to push on now because of time constraints. So our next speaker this morning is Dr. Ikabonga Mukumbuta from the University of Zambia. Ikabonga worked as a postdoc on a second project, which was allied with CEPHaS. It was part of our plan to apply for additional funding, and we were successful in getting a grant on the history of soil surveys. A little bit unexpected, perhaps, for soil scientists to work with historians, but we think this is an important kind of cross-disciplinary area to develop. So Ikabonga worked as a soil scientist, but with historian colleagues on this project. And what he's going to talk about is legacy soil information. Legacy soil information is a framework to address contemporary challenges. And this is in particular a perspective from Zambia and legacy information held there. So thank you, Ikabonga. I'll hand over to you.
Thank you very much, Mary. I hope you can hear me. Okay, great. Yes. So thank you very much for the introduction as well. So my name is Ikabonga from the University of Zambia, and I'm going to present on behalf of my colleagues from the Instanza project. And we are looking at legacy soil information as a framework to address contemporary challenges, and I'll be presenting the perspectives from the Zambian context. Next slide. Yeah, so I hope that most of us in here appreciate why soil information is important, and it's particularly important because it can be used for research and development and for decision making at various levels. So, for example, if you're looking at what kind of crops should be grown in which areas, which areas, for example, you can open a new farm, or whether you are looking at irrigation appropriateness in different fields, but also in terms of ecosystem services. So, for example, what is the soil carbon content of particular soils, and therefore from there you can try and estimate the carbon sequestration potential of the soil. So these are just some of the questions that soil information can answer, which can help in terms of decision making. Next slide. And in Zambia particularly, and I think in most African countries, there's increasing demand for soil information. And obviously this is because there's this agenda to increase productivity, especially among smallholder farmers. And therefore soil information is needed for decision making at that level. But availability of soil information is limited, and it's largely hampered by the very high cost to conduct soil surveys. Okay, so it's very expensive to conduct soil surveys. However, we know that there is some soil surveys that were conducted, for example, pre-independence by colonial governments, but even immediately after independence. Okay, so in this project, we wanted to find out whether this legacy soil information is useful today to different stakeholders, and if it is this information accessible to various stakeholders such as farmers and researchers, so that then they can utilize it for various decision-making processes. And then also we wanted to find out the quality. I mean, if there exists this legacy information, is it of good quality to be used by the various stakeholders? Next slide. Yeah, so in terms of the materials and methods, so we focused on the eastern and southern provinces of Zambia. And this figure shows some of the points where these legacy soil surveys were conducted, and the scales. So generally the scales range from 1 to 50,000, 1 to 10,000, in that order. So in terms of the process that we did, so the first thing was for us to do an inventory. First of all, of the surveys that we know were conducted. So at the Zambia Agricultural Research Institute, ZARI, is the primary board that produces this information. So there's an index of, or a list of the legacy soil surveys that were conducted. So then we did so once we have that information, then we tried and find these reports and maps. And then once we did that, we developed, please go back to the previous slide. And then we developed a protocol for reading and extracting this information. And then the third aspect was evaluation of this extracted information. And then finally, we had a stakeholder workshop to assess the utility of this legacy soil information. And the first two, I think, are pretty straightforward. So I'm going to explain briefly on the evaluation aspect in the next slide. Yeah, so for the evaluation of the quality of the extracted information, so we use the Cornell adequacy protocol. So this protocol lists, parameters that you need to test to assess the quality of legacy soil information. And the first parameter that we chose for analysis was average size delineation, which is basically shows you the average size of the polygons of the map. So, or in other terms, the average size of the map units. And so basically for this one, if you have a legacy map like this one shown here, then you can make a circle of non-radius, and then you can count the map units that fall within that radius. And then from there, using this equation, you can estimate the average size within that particular area. Next slide. The second parameter that we used was the index of maximum reduction. So as you'd imagine, when you produce a map, there's a minimum legible delineation. So there's a minimum size that can be legible on the map. So we use that factor, and also and we divide the previous information, which is the ASD, by the minimum legible delineation. So this gives us an idea of how legible are the map units on that published scale. The next slide. And then the last one that we use for evaluation is the boundary density, which is basically the mean length of a map boundary per unit area of a published map. And so this one, briefly, if you have 2 points with and these are randomly generated points with non-coordinates, and then with a non-distance between them, then we can count the number of map units that intersect with that line, and then from there we can calculate the average boundary density of that particular map. Next slide. And so once we did the evaluation, we did the inventory, and then we developed the protocol and read the information, and then we evaluated that information. Then we had the stakeholders workshop, which because of COVID, was done virtually. So during this workshop, we presented to the attendees from various institutions information on soil, on legacy soil information, but also just some of the contemporary soil information. And so after the presentations, we administered the structured questionnaire, and then they sent back those questionnaires, and then we extracted the responses in terms of the utility of the legacy soil information. Next slide. Yeah, so briefly, the results. So in terms of the availability and accessibility of legacy soil information, so our evaluation found that in the 2 provinces, 57 surveys are listed to have been conducted during the period under review. So in those 2 provinces, the Eastern Province and Southern Province, a total of 57 surveys or legacy surveys were conducted. From that number, we could not trace or find reports or maps of 64% of these surveys that are reported to have been conducted. And I must mention here that we could not find these at ZARI, which is the Zambia Agricultural Research Institute, and the University of Zambia libraries. So it's possible that they could be available elsewhere, for example, maybe on people's desks, in some offices, but not at the 2 libraries, which normally would have this information. And of these, only 22% had both the map sheets as well as the survey memos. Yeah? So meaning therefore that the previous slide, please. And then from these, only 14% of them only had the memo, so we could not locate the map. So this shows in terms of the limited availability and accessibility of this legacy soil information. Next slide. And this is just an example of one legacy soil information, which is the Chipangali State Farm, which is in Eastern Province. And so you can see here just a scan of the memoir. Next slide. And here, so this is the kind of information that we presented to the stakeholders. So this shows, for example, the map. And then if we focus here, of course, we can get the area and the scale and so on, but we can also focus and see the map units that were there. And for example, if we focus on map unit 2, then we can extract from the legend what kind of information is available from the memo. So for example, yes, next slide. Yes, so from the legend, for example, here, there's a description there of that map legend for unit 2. So it gives, for example, the depth of the soil, the color, the slope, the texture. Next slide. And then we check the survey memo to see if we could get extra information. And here we could find, for example, some chemical data such as the cation exchange capacity, the pH of the soil, and other information. And then we asked the participants whether this information was useful to them. Next slide. In terms of the assessment of the quality of the surveys, so this table shows you some of the legacy soil surveys. So on the left are the names of those surveys. And then we have the map scale there. We have the boundary density, which is D. Average size delineation here, ASD. And I want you to focus on the right side of which is IMR, which is the index of maximum reduction. And then at the bottom here, we have the optimum value based on the Forbes protocol. So the optimum value for the index of maximum reduction is supposed to be 2, okay? And if you look at all our surveys, this information, you find that the IMR for almost of them range from about 3.3 to 7.4, which means they are much higher than the recommended optimal value of 2. And this basically means that the legacy maps are legible, but their scales could be reduced to what they are, and without losing any legibility. So in short, this means that these surveyed areas are represented on a map that is physically larger than what was necessary. So this could have had implications on the cost of producing those maps, although the quality, of course, is good in this case. Next slide. I cannot see the next slide. Okay, no, the previous slide. Sorry, I think there's a lag between the movement of the slide and yeah. So here, again, the same surveys. And then here, this figure shows a map intricacy, which is the number of intersections of the map units per kilometer. And then if you look at the estimated, which is the calculated value from our analysis, and then on the right is the predicted value based on the map scale. Okay, and you can see here, if you look at the top two surveys, Upper Kalea and Chabala, which were at the scale of 1 to 10,000, see that the calculated value here is lower than the predicted value. Okay? So this means that for these at a larger scale, the number of map units that were in that map were fewer than what was expected at that scale. However, for most of the surveys, from the 30,000 scale and the 50,000 scale, the calculated map intricacy was very close or almost the same as the predicted value. So at these scales, it means that the number of map units here are within what was expected. So in general, meaning that the quality here of these surveys is acceptable or good. Next slide. And this slide shows the stakeholders that took part in our workshop, and also sent back the questionnaire. So briefly, we had a representation from extension services and research arms of the government. We have national and international NGOs, people from academia, private sector, large-scale commercial farmers, and also private sector in terms of agricultural service providers and those who provide input, and also research and development divisions. And that just shows you the representation as a percentage of those that responded. Next slide. Yeah, so this table here summarizes the views of the stakeholders in terms of the utility of the legacy information. And briefly, the kind of questions that we're asking, for example, are what aspects of the legacy information was of value to the stakeholders, and then what they think were the reasons that the soil information might be of limited or no value based on the presentation. And then also what the main limitations in terms of value of the contemporary soil information was to them. So these are the responses that was given, but I'm just going to highlight, if you can click next, just briefly summarize the many responses. So generally, almost all stakeholders did indicate that legacy soil information was very important and of value to them in terms of decision making and planning. However, there were a number of misgivings, especially as related to this legacy soil information not being readily available or accessible for them. There are also some complaints, especially for non-technical, especially farmers, that the soil information is perhaps in a less user-friendly format, and they'd like it to be a bit more user-friendly for them. And also another issue was that sometimes the location of where these surveys were conducted was not really representing where these people were coming from. So the legacy soil information needs to be location as well as use specific, depending on which stakeholder and what kind of information that they are looking for. Next slide. Yeah, so briefly, that was our study. And in terms of the conclusions, so the main conclusions are that generally we found that this legacy soil information, though it was done, is not readily accessible, at least at the libraries that we went to. Although, of course, we must mention here that it could be present somewhere, but just not accessible enough for us to see. And also we noticed that the repository for this legacy soil information is generally not well organized. So it wasn't very easy to, for example, locate a particular memoir or map that we are looking for. And therefore, this means that there is great need, based on the response, that this information is valuable. There is great need to preserve this legacy soil information in terms of, for example, digitizing this information and, for example, maybe putting it online where anyone can easily access it, but also making sure that there are copies, either digital or hard copies at the different national libraries, for example, the University of Zambia, ZARI, and even at the National Archives, so that many more people can have access to this information. In terms of the quality, generally, the legacy soil surveys had good quality, so that means we could extract useful information from them. And stakeholders did indicate indeed that the example information that we presented was useful and it was of value to them. Although there are issues in terms of availability, as well as the need to make this a bit more user-friendly or in a format that can easily be consumed by the various stakeholders. So that is all. Thank you very much for your attention.
Thank you very much, Chikoye, for a very clear presentation. I don't think we've got any immediate questions, but there will be a chance for people to share any questions they have later in the chat. But I think there was a very interesting complementarity between what Chikoye was presenting previously on using the quantitative information directly, and then what you were presenting in terms of the soil map units, the classes, which represent so much user experience or surveyor experience in the field. So thank you very much for a very clear presentation. And as I say, if anyone does come up with comments that they would like to make to Chikoye, questions, sorry, to Chikoye, please do put these comments into the chat.
Hello, everyone. Thank you for sticking with us. We are going to start again with our program for this second half of the day. And we are going to be introducing our next speaker. And I shall pass over to Murray to introduce.
Thank you, Alasia. So our second keynote speaker today is Dr. Matthew Mbanga, who is CEO of Foundations for Farming. Foundations for Farming is focused on advocacy for transformation of land use, trying to come up with more sustainable systems whereby individuals and communities can enjoy a better quality of life through improved food security and management of their land resources. So we're very grateful to Matthew for agreeing to give a keynote talk today, and very much look forward to hearing his talk. And he's given us the title of More Yield, Less Field. So I shall hand over to Matthew.
Good morning, thank you. Sorry, having a bit of trouble there with my sound. So sorry, I won't be sharing my, I won't be putting my video on. I'll share my screen though. I'm holed up in my bottom room, having tested positive for COVID. And my wife was saying to my son, you can't go down there. We've got a COVID person in the house. And so my son said, but I love that COVID person. So I'm sitting here by myself, holed up, and I'm hoping that I will be able to get through this without too much spluttering and sneezing. Okay. So I'm going to be speaking to you about more yield, less field. This is kind of the program that we've run here in Zimbabwe under the name of Mukumbuta, which is our conservation agriculture approach that we've introduced in Zimbabwe and run successfully really for the last 10 years under that name, and kind of trying to introduce conservation agriculture and make it applicable to small scale farmers in Zimbabwe. So we've been on a wonderful adventure as Foundations for Farming really for the last 40 years. I've only been involved for the last 11. But the founder began this journey, a guy called Brian Oldreeve, began developing sort of minimum tillage and kind of these climate smart. What he didn't know then was this climate smart approach to farming in Zimbabwe, or then Rhodesia, back in the late 70s and really started to really perfect it and kind of gain momentum in the 80s. He was a large-scale commercial farmer in 1982, growing 1,000 hectares of row crops, so maize, wheat, soya, and losing money. So he was surrounded by a bunch of formerly white commercial farms where there were seed maize growers, and a lot of them were losing money, were going out of business, and it just wasn't financially viable to continue with conventional agriculture. Remember, this is a point when there used to be the World Championships of plowing, and Brian was one of the best plowers. He used to plow deep. You know, conventional wisdom was the deeper you plow, the better farmer you are. And so he was very much in that kind of thing, and really just realized that he was losing money hand over fist, and it was not sustainable. It was no longer profitable, and a lot of the farms around him went out of business. So he started trying to really just develop and research and kind of delve into this whole idea of minimal soil disturbance. He had heard about the Dust Bowls of Texas, but remember the early 80s, this is before Google, and he had no real opportunities to research or to kind of to find out too much about that. But what he did realize was that it was the plowing process that was at the root of a lot of his problems. So he started to experiment just initially on 2 hectares with not plowing and with covering the ground with a thick mulch blanket of crop residues and what we now would call mulch. So as he began that adventure, it took him 12 years to grow that farm, and his initial trial from 2 hectares the first year to 10 to 50 to 100 to 200 to 500 until the whole 1,000 hectares was being grown under the system and making profit every year for 12 years. That led to him being so successful and profitable that he established a national maize yield record that stood for 20 years. And in that sort of time as well, they acquired or purchased 5 surrounding commercial maize seed maize farms that were all going out of business doing conventional agriculture. So he realized clearly that he was onto something, had this incredible breakthrough at scale, grew that farm from 1,000 hectares to 3,500 hectares, the second biggest contiguous maize or row cropping farm on the continent of Africa. That's in about 1995 by that stage. So then at that point in around 2000 or late 90s, the Mugabe regime brought in its land reform program, and all of that land was mandatorily acquired by the government, and Brian was kicked off. And that began his journey really with small scale farmers and serving into the most vulnerable communities in the nation of Zimbabwe, because we were unable to feed ourselves as a nation from that point onwards. So agriculture in Africa and sustainable livelihoods is not a kind of a new thing. Africa has always kind of relied on the land, and 70% of Africans, not only in Zimbabwe, across Africa, rely on the land for their livelihoods. And so 70% or 700 million people on the continent of Africa rely on agriculture. And yet it's not really a profitable thing. It's not seen as something sustainable. It's not seen as something that is noteworthy, or there's no dignity. There's no kind of excitement about entering agriculture. As I speak in schools and I work with young people and I ask what they would want to do with their lives, it's zero. Not a single person puts up their hand to say, I want to be a farmer. I want to work the land. I want to grow food. I want to be on the land. So that's a great tragedy. What Brian discovered back in the 80s and 90s and perfected at scale was the understanding that yield is directly proportional to standards. So the standards of agronomy that we apply to the soil directly affect the yield that we're able to take off. So as I said, when there is this kind of lack of dignity, lack of professionality, education, purpose at small scale farming, despite the fact that we rely on the soil and the land for our livelihoods, it's not very productive. It's not done, it's not treated as a science, it's not done to a very high standard. And so as a result, we have very poor standards of farming and poor standards of agronomy throughout the rural sort of areas of Africa, certainly here in Zimbabwe. So these poor standards equate to poor yields. Here in Zimbabwe, less than 1 ton per hectare, which is not even able to meet our household food requirement. In a subsistence environment, Farming was not done as a business. It's only left to the elderly, the young, and the infirm who are left in the village to grow the food. And partly, I suppose, as a bit of the resource curse here in Zimbabwe, you know, we've got great climates, great soil, great rainfall, all those sort of things. It didn't take a lot to just produce a basic amount of food. And so when it was done as a hobby, when it was just done kind of at a subsistence level, that was fine. Because we had commercial agriculture, we had this massive agricultural powerhouse that was feeding us and the region. And Zimbabwe was the breadbasket of Southern Africa, based sort of on the back of those large commercial operators. But following Mugabe's mandatory land acquisition without compensation exercise in the 2000s, Zimbabwe became a net food importer. And overnight, the economy collapsed with soaring hyperinflation. For the last 20 years now, since that time, Zimbabwe has relied on food grown in the village or donor handouts. So now those small subsistence farms dotted around the country, we now actually need them to produce food. The Minister of Agriculture told me last year, during his visit to our training center, that the government of Zimbabwe was spending $30 million United States a month to import basic foodstuffs. So that would be your maize meal, which, as we've heard from a previous speaker, is our subsistence diet. Here in Zimbabwe, our staple diet is ground maize or corn. So in order to transform the nation's food system and turn impoverished rural farmers into the food producing machine that we require to feed the nation in the absence of large commercial producers, our mission as Foundations for Farming was to equip a rural base of largely uneducated, largely female, young, old, or infirm farmers into a dynamic, profitable, and sustainable base of consistent, decentralized productivity for food. In order to be effective, we knew that these plots had to be small. Most of these farmers don't own a lot of land. The average size for these small-scale farmers in Zimbabwe is about 1.8 hectares, which is sort of 2.5 football fields. Okay, it's a fairly decent size of land, but in terms of agriculture, we would consider those to be small-scale farms. So the average harvest from that entire 2.5 football fields per farming family is less than a ton. At the same time, it's heavily reliant on synthetic fertilizers and plowing, so ox-drawn or traction plows, causing massive environmental degradation, moisture loss, erosion, topsoil loss, and the silting up of rivers. So the methodology needed to be simple, it needed to be low tech, it needed to be straightforward, it needed to be manageable in terms of the land size, it needs to be applicable. So we use hose and we use this kind of climate smart sort of response, obviously with maximum soil coverage. So that's your mulching. It needed to be long lasting, which means it needed to be low cost. It needed to be accessible, it needed to be sustainable. And above all, it needed to be regenerative. So the 4 principles that we've kind of used as Foundations for Farming. And so we've spoken, I think I saw a little bit of this in the chat as well, about the 3 pillars of conservation agriculture. We've added a 4th one to that. So minimal soil disturbance is really, really well understood in terms of protecting the soil, increasing the kind of the organic material in the soil, as well as feeding kind of the microscopic organisms that live in the soil and create the channels for water infiltration that create natural fertility in our soil. So we want to protect those guys. So minimal soil disturbance ensures that the soil profile remains intact, that all of those microbes can remain alive, both the aerobic and anaerobic, can stay there in place and do their job. And then, of course, your mulch, so that the crop residues that are left on the soil surface and covering the ground, or grasses and leaves that are brought in to cover the ground and to allow the rainfall to infiltrate, to keep moisture in the soil, as well as to break down and do everything else that I've just explained. The 3rd one, in traditional kind of an understanding of CA, is your rotation, so what we would call mixed cropping. Just so that we get the 4 M's. But your mixed cropping is really just a hard rotation. So every season we rotate with a legume. Obviously, in terms of just good agronomy, you want to continue to maintain that healthy soil sort of makeup with your nitrogen fixation from the legumes. But also you want to break the cycle of diseases. So by rotating every season, we bring that in. And the fourth one, which is the one that we've added, is management. We want people to be able to manage. Again, standards are directly proportional to yield. So we wanted people to be able to manage their plots well. This involves disease scouting, scouting for pests. It also involves being able to weed. So high management is critical. It's a critical part to this whole process. So in order to achieve this small, simple, accessible, manageable, long-lasting impact, we asked the question, how much food, and we're talking about maize, does it actually require or does it take to feed a family of 6? And so the answer that came back to us from that research was 1 bucket of shelled maize. So a bucket is typically a 20-liter bucket. And so we filled that up with shelled maize and weighed it. It actually weighs 17 kilograms. So you can see in the picture, these are some of our staff when we're just doing our trials. And we're working out exactly how many cobs of maize do we have to harvest in order to fill a 20-liter bucket. What we found out was that we need 56 cobs. So we could do 28 planting stations with 2 plants per planting station. That will give us 56 cobs. Each of those cobs it's got to be a decent sized cob. So those are about 300-gram cobs. And if we had 56 of them, that will fill a 20-liter bucket of maize and provide 17 kilograms of shelled maize. So that's a week's worth of food. With our spacings and the spacings that Brian, the founder, had kind of perfected at scale, that meant a 60 centimeter in-row spacing, yielding a 16-meter row. So one 16-meter row provides a week's worth of food. Now there's 52 weeks in a year, so we need 52 rows to provide 52 buckets of food every year. So the final measurements of the plot are 16 meters by 39 meters, or 1/16 of a hectare. That's about the size of a tennis court. So the average yield from this tennis court size of maize is 850 kilograms. And if we extrapolate that out to tons per hectare, that's the equivalent of 14 tons per hectare. That was the national average, the national record, that Brian set way back in the 90s. 14 tons per hectare equivalent, simply by following this simple, manageable, accessible, long-lasting approach to growing household food security. So remember, this is against the national average of less than 1 ton per hectare, which had become the average by 2019. So this is achieved by making it possible for farmers to implement high standards of conservation agriculture. They can implement the 4 M's that we've just spoken about: the minimal soil disturbance, the mulch, the management, and the mixed cropping. So our heart is very much to go to a kind of an organic, no reliance on pesticides and fertilizer and all of that sort of thing, to be truly regenerative and truly sustainable. But we know that it's just too big of a jump to get farmers away from conventional agriculture to what we're doing is kind of a big enough step. So we've stopped short at this point of sort of making it completely organic, but that's certainly our heart, and it's certainly where we're going. We certainly teach that to our farmers who are compost making and so on. But in order to make this a kind of an easy enough jump for high adoption rates across the country, we've come up with a simple sort of a mantra: on time, at standard, without waste, with joy. And so this addresses all of our issues that we're facing. So the science, it's on time. We've got to be on time. Now, if we plant at what has become the national average planting day, which is the 22nd of December, we worked out that for every day that we plant after the 25th of November, which is the optimal planting day in Zimbabwe because of the available heat units of the sun here in the Southern Hemisphere, we lose 120 kilograms of yield potential per hectare per day. That means that if we plant as we have been for the last 20 years, on the 22nd of December, before we even put seed in the ground, we've lost 3.6 tons per hectare of yield potential simply by planting late because we've missed out on the available heat units. Now, by having the mulch cover in place and by being able to, by not disturbing the soil, by not increasing the surface area of the soil for evaporation and all those sorts of things, even with climate change, we're able to be fairly successful at gaining this crop and being able to carry the crop through and not have moisture stress by planting as close to the 25th of November as possible. So this sort of system has become known as climate-proof conservation agriculture. So it's climate proofed because it's fail safe. Because normally, certainly here in Zimbabwe, by about the end of November, we should have reached a cumulative amount of rainfall of about 100 millimeters of rain, which is an effective rain, certainly for the maize crop. So that's the science kind of element of it. The longest day of the year, the 21st of December, is the same every single year. It doesn't change. So we must be on time, and we help farmers to prepare on time and to be able to be on time, to do things at a high standard. This addresses the need for good agronomic principles. So the agronomy of being able to do things at a high standard, that means your rates of fertilization, that sort of thing. It's so much easier if you've got these straight rows. Your spacing is critical for your population densities and those sorts of things for your crops. You can achieve that by having high standards, by using terrain ropes to measure, by having your correct inter-row spacing. You can move up and down within your crop. You can scout for diseases and so on. This plot is actually so small as well that if it doesn't rain, or if there's a mid-season drought, as we've become accustomed to, you can actually water it by hand. So the standards are imperative. And there's also a lot of dignity and joy that comes when you see a farmer with straight rows. They want to stand next to their field. You see people standing there with their hoe in their hand, being so excited to be recognized that they're the ones that have done this. And we love sort of visiting the farmers and seeing that real dignity as the farmers stand beside their field. And of course, the yields then kind of match up to that as the standards equate to high yields. So without wastage, we don't waste time, we don't waste our labor, we don't waste our inputs. All of the inputs go directly, it's precision farming, they go directly into the hole, so there's no wastage. And then your profitability, of course, because of that, is through the roof. And then with joy, you know, we help people because of these high standards and because of kind of the transformed sort of thinking about productivity and about farming and about food production. There is joy, and these farmers, then that becomes the work ethic. People want to be in their field because they feel that purpose and that dignity that comes from doing things well, and by following all of the other principles, doing things on time at a high standard, without wastage and with joy. So my final slide is about regenerative agriculture and food sovereignty. We partnered with our government last year, and the government of Zimbabwe was targeting 2.2 million farming households in the country. We called the program Mukumbuta, which means new season. And so Mukumbuta is really a kind of a prophetic sort of a thing of saying that we are ushering in this new season in Zimbabwe. And so we were able to do that and train 13,600 extension staff, community-based trainers, leaders, agricultural college staff and farmers in all 10 provinces and 56 districts across Zimbabwe last year. In total, this program targeted 2.2 million small-scale farming households and 9 million beneficiaries. The result of that in 2021, this year, was that Zimbabwe harvested 2.71 million tons of maize. 40% of that total yield or harvest was grown by small-scale farmers, and it is the highest yield in Zimbabwe since 1984 at the height of our agricultural prowess. It is the third highest yield of all time in Zimbabwe. And in August, Brian, the founder, received a presidential commendation from the president of Zimbabwe and has been credited with making Zimbabwe food secure for the first time in 20 years, using this simple, manageable, applicable, long-lasting method of conservation agriculture that has brought about tremendous profitability through more yield and less field. Thank you very much.
Thank you very much indeed, Mati. That was an extremely interesting and inspiring presentation. I'm not sure we've got any immediate questions in the chat. One I would like to ask you, one of the issues that has repeatedly come up throughout the course of the CEPHaS project is weed control. And with your aspiration to promoting fully organic systems, I was wondering what your views are on how weed control is best achieved without tillage.
Oh, yes, so the mulch is a huge part of that. Maybe if I can just return to one of these slides. Are you still seeing my screen?
Yes, yes,
so,
you can see.
So you can see the crop residues between, this is actually a soya crop that you can see here. And so you've got our maize yield again with the rotations. You've got the previous year's maize residues on there. And so that is really the greatest tool that we have in terms of weed control. So the mulch kind of inhibits the weed growth, and it makes it very easy to pick off the weeds. Any weeds that kind of do grow and come through that, we can just pick them off. You can see there is still quite a lot of bare ground visible here because this is early on in the season. We would kind of come back in and backfill and bulk that up as the crop grows. And then of course the canopy of the crop itself will also shade out, and that's why your crop spacing is so important. The crop canopy actually shades out and inhibits any further growth. So we've now been farming on this particular plot of land here that is our training center for the last 8 years, and we have virtually no weeds now. We've broken the cycle. We've kept on it. We work very hard with our weed control, and we've stayed on it. And so we've now removed all of the weeds, and we never allow them to go to seed. So there's no new seeds being seeded or grown in our fields. So for me, the mulch is one of the greatest kind of assets that you have in terms of weed control.
Thank you very much indeed, Matthew. And as I say, it was such an interesting talk, and really showing the potential of rethinking some of the ways that farming is done.
Yes, sir. Thank you.
So thank you again. Our next speaker now from the CEPHaS team is Dr. Elijah Phiri from the University of Zambia. And Elijah is going to speak on behalf of a large group of CEPHaS team members, mainly from Working Group 1 in soil physics. And the title is Modeling Soil Hydrothermal Regimes under Reduced and Conventional Tillage Systems. So Elijah, hand over to you.
Yeah, thank you, Mary. Let me just share my slide. Are you able to see?
Yes, thank you.
Oh, okay, thank you. Yes, I'm going to present a preliminary analysis of one of our sites here. I'll look at modeling soil hydrothermal regimes and reduced and conventional tillage system. Just to give some background, we all know that there's limited information which is available, and that normally on conservation agriculture, especially that relating to soil temperature regime, as well as distribution of water within the soil profile, which supports crop growth. We also know that there's scarcity of water resources, and it is important that we look at increasing food production, food demand, and this is exacerbated by the increasing population growth and rapid economic growth. So normally we have software packages which can be of complexity at different levels, but available, and we can use this to do a simulation of water and heat movement in a soil plant atmosphere. And this will help us estimate some indicators of how useful it can be in terms of increasing water use efficiency. So one of the tools is the Hydras program, which is a numeric program which tries to solve Richard's equation for unsaturated as well as saturated flow. And also it uses, it has some components of heat and solid transport. So the objective of this study was to assess these soil hydrothermal regimes and water energy balance under CA and conventional cropping system. And in this case, we applied the Hydras 1D model. In terms of materials, we had to install sensors as part of the CEPHaS research team. We had sensors which were installed so that we have in situ monitoring of soil temperature as well as soil moisture at some of the depths in the profiles. In this case, it was 10, 20, 40. So the first one, 60 and 100. And also we do have a weather data which is generated from an installed automated weather station on site at the site. So this is at Riempe, where we have an experimental plot where we have 2 treatments with the soil maize under conventional tillage system and a maize soybean intercrop where residues are maintained and with the minimum tillage. So we are monitoring a number of parameters, but for this study, we are looking at soil moisture and soil temperature. And the plots are 50 meters by 25 meters. So by looking at just the general properties of the surface soil for this site, we do have low pH of 4.9, 4.49, as well as if you look at the retroconductivity and organic matter quite low of 1.3%. And as well as total nitrogen of just 0.19. So these soils are quite low in nitrogen, as well as organic matter content. And the soil texture for the surface is a sand loam with 60% sand and 30% clay, and the silt just around 9%. So these soils are highly compacted, just so even at the surface, we already have a 1.65 grams per cubic centimeter in terms of compaction. The available water holding capacity is close to about 180 millimeters per meter, basing on a field capacity and the permanent wilt point of this soil. So we have a study where we monitored these parameters, and we first tried to look at the data in terms of weather on site. We decided just to use, because we are doing modeling, we say pilot analysis for a very long set of data, which spans from 2017 up to now. So we decided just to extract for 3 months to apply Hydras in that period to look at its predictability. But on this site, from the weather station, we have daily temperature, which is below there, the ambient temperature, and the soil temperature, which is a sensor installed at the weather station. You have that almost close to 2-degree difference of ambient temperature and the sensor installed at about 15 to 20 centimeters in the soil. So we have slightly high temperature in the soil as compared to ambient air temperature. And during the same period, we have rainfall which fell, and we'll see how this affects both temperature and so in the soil profile. First, if we look at the effect on soil moisture profile versus the conventional tillage and conservation agriculture, we see the first one, the conventional one, that we have a drastic drop during this period of our data set of moisture content in the top 10 to 20 centimeters whenever, when the period when there wasn't enough rainfall. But if you compare with CA, you see that this is drastic fall difference between the deeper layers and the surface layers is not as prominent like under CV. So you're finding that there's a higher moisture loss in the top surface layers of the field, which is under conservation, under conventional tillage as compared to CA. But also worth noting is both ends is that the moisture profile at a deeper depth of 100, there isn't much change, but you see that the influence of rainfall goes as deep as 60 centimeter depth. Whenever it rains, you have a reflection of that as compared to the other depth. So in terms of moisture, we see that there is that difference between conventional tillage and conservation tillage. Again, when we look at soil temperature, we note that in terms of variation, the blue one being sensors installed at 10 centimeter and red one at 20 centimeter, we note that the effect is much warmer under CA. As you can see, the red part, that the variation and even the magnitude of temperature goes as deep as 20 centimeters as compared to under conventional tillage. So there is definitely something what is happening, because it's showing that the CA plots are much warmer than the conventional tillage plots. So it means that the structure and maybe moisture and so on might contribute to this. And so the CA plot seem to be warmer than the conventional tillage plots, at least up to a depth of about 20 centimeters. Again, when we put the data together for all the data for the entire period of 2 months, we see that in terms of the soil temperature profile, under conventional tillage, the temperature is lower as compared to CA plus. It's a higher temperature, and that explains even at 20 centimeters, we saw that it's quite a high temperature at 20 centimeters. So this, and the same, if you look at moisture in the profile, you'll see that the conventional tillage has a lower soil moisture content as compared to the CA plus. This might contribute to this warmer because water has a higher thermal heat capacity. So the fields or plots which are under CA tend to be warmer. This is what the results are showing from this. Then we did some modeling of the data. For this modeling, we just looked at part of the data set. So we looked at for 10 days. We see that when we model this being rainfall events during a 10-day period, and for example, we can pick on the green being the measured, and the blue being predicted at the same depth. So we see when we look at that, the trends in the modeled versus measured tends to be following almost the same trend in terms of temperature at all the depths. However, we are yet to fine-tune the model so that this difference between measured like this green and blue are almost close to each other. So we are working on it to improve on the prediction. So this is under conventional tillage. The same behavior is with a CA approach that we are able to predict. However, there is that small difference in terms of the temperature as we do the prediction. Similarly, if we look at where we had a bit of challenge with the model predicting, it is oversoil moisture where we need to fine-tune the model so that it predicts, because this on top, that is the predicted soil moisture at different depths, while these lines below, they are measured. So there is this gap difference. So we need to look at the fine-tuning of HADRAS so that the prediction, but in terms of the trends, are all reflecting the rainfall as well as you see the peaks are almost similar trends in the, but the gap which is there, we need to rework and fine-tune the HADRAS so that it is able to predict similar to what we actually measured. This is under conventional. Then again, under conventional agriculture fields or plots, the same trend is observed in our modeling, but there is this gap. So we are working on this so that we improve on the prediction of soil moisture, and as we compare it with the predicted. So looking at this data set, we can conclude that the soil profiles under CA had consistently higher soil moisture and higher soil temperature than under conventional tillage system. Again, we noticed that the soil moisture content of profile under CA, the surface layer was not lava, sorry, it was lower than under CV. Again, from these results, we see that the study indicates that the physical-based HADRAS 1D model is able to simulate trends in soil water and soil temperature. And we can do much more further analysis to look at partitioning of the different components of the energy balance as well as water balance. Thank you for listening.
Thank you very much, Elijah, for a very interesting and wide-ranging presentation. Do we have any questions? Right, so we have a question from, so I can't see all of the chat, from Isaiah Nyagombo, so saying that these are interesting results on soil temperature, saying that previous results from elsewhere have suggested that under CA, there's actually reduced temperature fluctuations. And his question was there a lot of mulch in the CA system? I seem to remember, actually, Elijah, that the temperature results from Dombashawa showed smaller temperature variations under CA, as Isaiah is suggesting. So why do you think it was different at the University of Zambia farm, at the MP?
Yeah, thank you, Mali. I think the reason is our field do not have as much mulch. So I think the buffering capacity of of temperature is more related to the soil structure improvement than the mulching. So that's why maybe the results are, but also this is a heavy clay soil. And I think that's how I can look at it. The mulching is quite low on these soils, on this field. In fact, because of termite attack when we harvest and so on, we find that by the time we plant, we have less stover left in these plots where we have not plowed or tilled.
Thank you. So I don't think there are any other questions coming in for Elijah, but we're on perfect timing to move on to our next presentation. So thank you again, Elijah. And our next speaker is Dr. Charles Chisanga. Charles is at Copperbelt University at Chikoye in Zambia, but prior to his move to Chikoye was at University of Zambia, and he's been part of the CEPHaS team in Zambia since the beginning of the project. Charles is going to talk to us about long-term trends in weather data, rainfall and temperature over Southern Africa, focusing on the three sites in the CEPHaS project, Chitezi, Dombashawa, and Liempi. And he is presenting on behalf of Elijah, Chwezi, and Nyanda. So, Charles, can you hear us?
Yes, I'm getting you.
Great.
Okay, are we happy to start, Charles?
All right, thank you. Yes, we can start.
Brilliant, thank you.
All right, so our discussion is, our presentation is on long-term trends in rainfall and temperature over Southern Africa, vis-a-vis at Chitezi, Dombashawa, and Liempi. We can go to the next slide. So if we look at the literature, in this background, we can see that changes in temperature over the last decade has increased, and these changes have impact on socioeconomic well-being, and the increase in air temperature and variability in rainfall have been observed in different parts of the world. And generally, rainfall and temperature are important meteorological parameters that determines the environmental condition and agricultural productivity. Then these changes in temperature and rainfall have a direct effect on evapotranspiration, the soil moisture regime, and the availability of water resources for plant growth. Next slide. So the near surface temperature has increased by 0.5 degrees centigrade in the last 50 to 100 years over Africa. And certain scientists have observed the trends also in annual averages, near surface temperature over Northern and the Southern Africa. And the mean land surface warming in Southern Africa is projected to exceed the global mean air temperature in all the seasons. So while there is a proliferation of literature on regional and global scale with regard to rainfall and temperature trends, there are limited studies that have focused on local impact. And yet it is at this scale that the impact of climate change are adversely experienced, or this is where we actually experience climate change. Next slide. So the objective of this study was to assess the long-term trends in rainfall and temperature at Chitezi, Dombashawa, and Liempi, and the dataset which we used was NASA Power reanalysis dataset. So the results of this study will assist in appreciating the current climate at the three study sites, and may also assist in developing appropriate adaptation measures in conservation farming. Next slide. So we know that the study sites for CEPHaS is Liempe, Chitedze, and Domboshawa, as shown on in figure 1. Next slide. So the data set which were used for this analysis is from 1982 to 2020, which represents 39 years. And this data was downloaded from NASA Power, which is the NASA prediction of worldwide energy resources. And this data set is point data, which can be downloaded using longitude and latitude for any specific location. And then the NASA Power data was evaluated using observations from Liempe weather station. We used the data from 2019 to 2021, and the statistics which were used to evaluate the NASA reanalysis data with the observations were the R-squared adjusted, the root mean square error, the bias, and the mean absolute error. And then the trends were analyzed using the Mann-Kendall test. But before we did that, we had to check the data for the first order autocorrelation at monthly and annual time scale. And the testing was done at 95% confidence interval. Next slide. So these are some of the statistics which were done between observed and modeled data. These are the monthly observed versus simulated maximum temperature at Liempe. So if we look at the R-squared adjusted there, we can see that it is 0.89, and the root mean square error was 1.64. And that also goes for minimum temperature there. We have a good R-squared, which is 0.97. And then for the mean temperature or average temperature, the R-squared adjusted was 0.96 with the root mean square error being at 1.5 degrees centigrade. But when we look at rainfall, we can see that the R-squared was 0.54, while the root mean square error was at 68.78. So we can see that the reanalysis data is able to reproduce the temperature values very well. Next slide. Next slide. So the next slide shows the daily observed versus simulated maximum temperature, minimum temperature, and the rainfall. If we look at the daily temperature, we can see that the statistics are very good, meaning that the reanalysis data is able to reproduce the maximum and minimum temperature. But when we look at the daily rainfall, that is where we have some issues. So when we look at the reanalysis data, at daily scale, they fail to reproduce very well the observed rainfall data. But when we look at the temperature, the reanalysis data is able to simulate the observational data. So these data sets, the reanalysis data, has been used by many researchers in different countries. For example, in Brazil, the NASA Power data set has been compared with observational data, and the monthly rainfall data showed good statistics, which with R squared being between 0.70 to 0.95 at 99 degrees confidence interval. Then the reanalysis data, which is being generated by various centers throughout the world, has been used in crop modeling, hydrological, agronomic, and also in climate change analysis. So high quality data, which is generated using satellite, is able to decrease information gaps where ground stations are scarce. So the reanalysis data can be used in locations and by scientists where we have scarcity of agro-met stations. Next slide. Next slide. So we also tried to plot the seasonal modeled data for rainfall and T-mean for the 3 sites. So if we look at the seasonal data, especially for rainfall, we see that Chiteze receives more rain in January, February, and March, followed by Riempe, then Dombo shower. But when we look at October, Chiteze still receives more rainfall, then Dombo shower, and then Riempe. But when we look at the temperature, for example, in October, November, December, we can see that Riempe has higher temperatures, followed by Chiteze, then Dombo shower. But in January, February, and March, we see that Riempe still has higher temperatures, followed by Dombo shower, then Chiteze. Next slide, please. So when we look at now the actual trends in the parameters, the results indicate that the annual rainfall showed non-significant statistical changes at all sites. So when we look at Chiteze, and especially the sand slopes at monthly and annual scale, so rainfall at Chiteze has been reducing by 4.04 millimeters at monthly scale, and by 6.75 millimeters at annual scale. Then at Dombo shower, the rainfall has been increasing at 2.81 at monthly scale and 1.62 at the annual scale. And at Riempe, rainfall has been reducing at 0.99 at monthly and 1.91 at annual. So if we look at the temperatures, we see that the monthly temperatures and annual temperatures at Chiteze have statistically increased. At a monthly stage, the maximum temperature has increased by 0.03 degrees centigrade, which is similar to minimum temperature, while the average temperature has increased by 0.02 degrees centigrade. Then at annual scale, maximum temperature has increased by 0.03 degrees centigrade, while the minimum temperature and average temperature have both increased by 0.02 degrees centigrade. When we look at Dombo shower and Riempe, only minimum temperature showed a statistical significant positive trend at monthly and annual scale at the rate of 0.02 degrees centigrade. Then figure 5, which is on the next slide, shows the graphs of how the parameters have been changing. So if we look at the first panel there, it shows how we can go to the next slide. We see how the parameters have been changing at Riempe. Then the next panel shows Chiteze. Then the last panel shows Dombo shower. So these graphs give us a visual impression of how things have been changing, whether being significant or statistically non-significant. We can go to the next slide. So in conclusion, the NASA power reanalysis data can be used in analyzing impacts of climate change at local or regional scale. The observed significant positive trend has implication on soil water content, crop production and management, and this calls for appropriate climate change adaptation measures vis-a-vis conservation agriculture and climate smart agriculture. And this concludes our presentation on trend analysis in temperature and rainfall. Thank you very much.
Thank you very much indeed, Charles, for a very clear presentation. I believe we have some questions. Trying to find it. Is there something?
There it is, sorry.
Right, so it's a question from Hakunawadi Swarai. He says, is the failure of daily rainfall modelling due to there being fewer data points?
All right, thank you very much. Mostly the reanalysis data, they use satellite estimates, and also they are simulated with ground stations. So where we have fewer ground stations, the simulation may not be as accurate as possible, especially as far as the rainfall is concerned, because it is one of the most difficult variable to simulate. Thank you.
Yeah, thank you.
That's a previous question, so.
Right. I wonder, oh, right, there's a question from Dan saying, could you comment on the positive temperature bias for the NASA product? Saying, is one land surface temperature and the other air temperature, for example?
Okay, well, maybe if Alessia can take us to that slide so that we can see. We can go back, I think, on the month 3. Again, let's go back.
That was the one you were on. Can't up.
Okay. Now the question.
So the question was, yes, whether you could comment on the bias that we can see there between the simulated and observed temperatures. And Dan was wondering whether one might be land surface temperature and the other air temperature. Were they strictly comparable?
All right, because these air temperature data sets. Okay. So if we are looking at, is it land surface and air temperature? Mostly, sometimes they can be comparable depending on what you are comparing it with.
Okay, thank you.
I think that's everything for now.
So yes, so I think, thank you, Charles. I don't think we have any more questions, but of course, if anyone does have any other questions they'd like to put to any of the speakers in the course of this conference, where we carry on Friday, do please feel free to share them in the chat. So we have actually, thanks to some very nifty presentation there by Charles, finished on time.
Yes.
So thank you to Charles for that, and to all our speakers for a very interesting set of talks that we've had today. I think it's given us some idea of the range of activities in the CEPHaS project, and the keynotes have also given us a really fascinating insight into the broader context within which we're trying to work in the CEPHaS project. So I should like to thank our keynote speakers again, Dr. Chikoye and Dr. Mbanga, particularly like to thank Matthew for speaking to us despite his case of the COVID, and we hope that he makes a swift recovery and is soon able to interact more freely with his family, because they clearly enjoy his company. So thank you very much. Thanks to all our speakers, and I'll hand you over to Alessia with a reminder for the rest of our programme later in the week.
Yes, so there is one last day to go. It will be Friday morning, starting at 8 a.m. GMT time. If you haven't registered yet, please follow the Eventbrite link for the Friday event and register for this as well. And on Friday, we will be talking about monitoring water in soil, crop and aquifer and capacity strengthening. We will have again presentation from our team, and we will have two keynote speakers again, external speakers from Amsterdam University. If you want to join us, we would love to see you on Friday morning. We will be collecting the Q&A questions and answers and put together a booklet for everybody, because we understand that obviously some questions were not answered today. And I will be sending out a feedback form as well at some point after the conference. I would really appreciate if this could be filled and returned. And on that note, I shall see you all on Friday. Thank you so much for joining us, and see you then. Bye-bye.
Thank you.
Thank you. Goodbye.
Bye.